Hi everyone, good afternoon. So, my name is Nabila, and I'm here to talk about the regulatory perspectives of combination products, which is a very rapidly evolving area because it is the intersection of pharmaceutics, medical devices, and biologics. So, these devices they have such transformative potentials, but given that they also have unique challenges that need a very tailored regulatory evaluation. So today, my presentation, I will be walking you through the emerging innovations, the preclinical and the clinical considerations, and the new tools such as the computational models and the AI that are advancing regulatory science when it comes to combination products. So, what are combination products? So, combination products, for example, like drug-eluting stents and antibiotic-loaded dressings, they combine multiple therapeutic components. So, this presents unique regulatory challenges as these products they essentially blur the line between drugs, devices, and biologics, and this calls for a very robust regulatory approach. And by understanding their complex composition, we can ensure that they meet the standards of safety and efficacy. So, combination products first emerged in the 1970s with early examples like antibiotic loaded bone cements. Regulatory authorities define them based on their components and their primary mode of action. That is what dictates the main regulatory pathway. So, it is important that we have a clear definition of what constitutes their primary mode of action in a universal scale so that there is no difference between regulatory bodies and no lack of clarity in how we determine their PMOA. So here we have some examples of combination products. So A is a drug-eluting stent, which is used in a cardiovascular application. So these stents, what they do is that they provide the structural support to the blood vessels while also releasing some drugs, and they enable the arteries, they keep the arteries from not narrowing down again. And in picture B, we can see that it is an antibacterial catheter, which is designed with an antibacterial coating that helps to reduce the reinfection risk during catheterization. And in picture C, we can see an antibiotic-loaded bone cement. It is used in orthopedic surgery, especially as a substitute for a joint, like for joint replacements, and it is used to reduce the surgical risk that there is post-surgery. And in picture D, we have advanced wound care sticker. It is an integration of antibacterial as well as anti-inflammatory agents to help promote the healing process and to prevent infection. And in picture E, we have an intracranial drug-eluting stent. So this one, it provides, not only does it provide the structural support uh, in the cranial arteries, it also gradually releases rapamycin to prevent restenosis, which is like artery re-narrowing. It uses a biodegradable coating and it dissolves after 28 days. So now we have emerging innovations and regulatory challenges. So some emerging innovations, they have their therapeutic advantages that rely on new and novel cutting-edge therapeutics. For example, tissue engineering products. So the scaffold-based materials, they support the cell growth for in when it comes to tissue regeneration. And bioartificial liver devices, they offer liver support for patients that are suffering from liver degeneration or like liver failure. So this increases their survival rate until they have the chances of finding an accurate donor match or like a liver transplant. And then we have chip containing smart pills. It is used when it comes to the real-time tracking of medication intake, increasing the adherence and providing data to healthcare providers, like the medication intake data. So, with such a complex nature of combination products, the significant challenges to regulatory bodies are surmounting, and this requires careful oversight to ensure safety and efficacy across all the components. Because the regulatory frameworks differ internationally within the US, EU, and other regulatory bodies, this involves rigorous testing and including a clinical evaluation and performance monitoring in the real world. So, looking at the regulatory review and the technical evaluation, They are critical for ensuring that the safety, efficacy, and the compliance of combination products are not compromised. So, in first we have the life cycle supervision, which involves an ongoing oversight to address the evolving safety and efficacy profiles, like post-marketing surveillance and periodic reevaluation that ensure that the product's performance is consistent with the evolving regulatory and clinical expectations throughout its entire life cycle. And the core focus, the primary evaluation criteria, is centric on safety, which is defined as the mitigation of unacceptable risk, and efficacy, which is measured as the ability to achieve a clinically significant outcome. So this dual-ended focus, it makes sure that both the device and the drug components, they function in tandem, like there is a synergy between them. And next, we have interdisciplinary product, which blend the regulatory challenges of drugs, biologics, and devices, and the unique primary mode of action pathways that are required to be taken. But they demand interdisciplinary approaches, like, for example, in products like drug-coated stents or like pre-filled syringes. And then we have device-led products. So when the primary mode of action is a device, evaluation prioritizes the device performance, such as the precision delivery, the biocompatibility parameters, and the stability, while also integrating the considerations for the drug component. Next, we have the general considerations. So first one is risk identification. So the first step to this, it involves identifying and evaluating the risk by conducting a very comprehensive risk benefit analysis. So this ensures that the potential safety concerns such as the off-target effects and toxicity through the extra drug components that we are adding, they are systematically mitigated. And by using a component that is approved prior, like for example a drug or device, that if it has prior regulatory approval, we can leverage the existing evaluation that makes it easier for us to streamline the regulatory pathway. So through this approach, we reduce testing that is redundant, and while also making sure that the safety and efficacy are kept up to the standards in the new combination. And for scientific evidence, for novel mechanism of actions or any other new indications, or a new set of target population, we must submit robust scientific data to substantially claim that the efficacy and safety is met. Like, for example, if we incorporate steroids into pacing conductors to reduce inflammatory response and energy demands, we must demonstrate it with preclinical and clinical evidence. And any new risks that are introduced by any modifications that are made, it should be addressed. Like, for example, in implants, we might add antibiotic coatings um, to mitigate the infection risk. But there is another new risk possibility of antimicrobial resistance, which again involves a process of risk management to take place and again post marketing surveillance. So, that is also something we should be taking into account. Next, what is the rationale for choosing the drug component? Like, for example, drug-eluting stent, we choose paclitaxel and rapamycin. Why do we do that? And what is the rationale for proving that this is the most efficient most efficient choice that we could make? So, for example, drug-eluting stents, they use paclitaxel and rapamycin to reduce restenosis, which is the re-narrowing of arteries after the placement. So paclitaxel, it prevents excessive smooth muscle growth, while rapamycin is an immunosuppressant, which halts excessive tissue overgrowth. So because these drugs are like locally delivered, they target at the site directly and they reduce the systemic side effects. Next, you have pain relief gels that include hyaluronic acid with lidocaine. So this provides both immediate as well as long-term relief, and hyaluronic acid improves the joint lubrication and also reduces inflammation. While lidocaine, it functions as a pain relief, for fast acting, and blocks the nerve signals. So together they offer a holistic solution for managing joint pain as well as improving mobility. And we have antibiotic loaded bone cement. It usually uses gentamicin or vancomycin, which reduces the infection risk in joint replacement surgeries. So they are primarily chosen for their strong activity against the pathogens that reside in the surgical site, like Staphylococcus aureus. And again, their delivery is localized, which ensures that the systemic side effects are minimal while their efficacy is high. So the primary mode of action is crucial as it guides the regulatory designation. If a product's main therapeutic effect is device driven, it is device led, even if drugs support its function. So it is necessary that we ensure accurate designation to help direct products to the correct regulatory pathway. Next, we have the preclinical evaluation, which is interaction of different components in combination products. So for drug additions and device properties, adding drugs to devices can impact their production, storage, and even the device's mechanical properties. For instance, heparin oxygenators are designed to provide anticoagulation, but they may reduce the mechanical stability of the device. So, if such a trade-off, is it actually worth the risk benefit? So we should evaluate that to maintain and ensure both safety and functionality. And device carriers and quality control. When devices act as drug carriers, it is important that quality control is essential. So, various production processes, the sterilization, the storage conditions, they must be consistent, and they must ensure that the drug activity and release profiles are consistent and there is no difference. So, an a very feasible example could be an antibiotic loaded bone cement, in which case uniform drug distribution and sterility are crucial to prevent infections while also maintaining the structural integrity. Drug eluting stents. This highlights the importance of local drug concentration. The stents release drugs directly to blood vessel walls, minimizing systemic exposure while delivering high local concentrations to prevent restenosis. So, it is important to achieve this balance, which is vital for their proficiency in cardiovascular treatments. And the key focus areas include drug density to determine the proper drug dose per stent and the total dose, which balances the therapeutic efficiency with safety. Also, polymer selection, which is choosing materials that support stable drug release as well as ensure mobility, and also the release characteristics, which can be studied in vitro or in vivo to minimize the toxicity risk. For qualitative, quantitative, and in vitro release of drugs and combination products, it's it is important in terms of delivery. So first we have the drug dosage justification, in which case we must balance the efficacy and the safety thresholds. Uh, uh, like in the wound dressings and bone cements, the drug dose should achieve the therapeutic effects without a risk of causing adverse reactions. And for design impact on release, the structure and material of the devices, like for example a stent or catheter, it influences how the drugs are released. And in this case, it is vital that we ensure consistent and controlled release. In vitro release studies. These studies assess drug stability and the coating integrity over time, which is crucial for maintaining the effectiveness of long term therapies. And for bioactive substance evaluation, products like orthopedic devices or heparin coated tools require confirmation of the drug's content, activity, and the efficacy to avoid underperformance or risk. Biological evaluation. So, this ensures that the combination products are safe and effective by assessing the interaction between their components and their biological system. So, first we have the ISO 10993 standards. It is the guideline for biocompatibility testing, but by adding the drug components, we can alter the test results. So, which makes careful evaluation necessary here. And for the mode of action and benefits, the mode of action and clinical benefits, it should justify any of the biological risks involved. This balance ensures that the therapeutic advantages outweigh the potential adverse effects. And for local and systemic effects, for example, in a stent or the systemic impacts, it requires additional safety studies if the dosage exceeds previously approved limits. For prior approvals, new combinations of devices and drugs they must be evaluated to ensure they don't compromise the standards of safety or efficacy when they are compared to each individual component. And contact time. For products with varying contact times, like implants or dressings, they need specialized risk assessments to assess their individual biological impact. Animal studies. Animal studies they play a critical role in evaluating the safety and efficacy before human trials. So, in the case of in vivo drug release dynamics and pharmacokinetics, they help predict how the product behaves in a biological system, ensuring the right performance and safety profile. For drug eluting stents, they evaluate the delivery performance, toxicity, dose levels, polymer coated thickness, and pharmacokinetics, ensuring the controlled release and minimal side effects. In silver contained wound dressings, animal models mimic the product's intended use, assessing the application methods, frequency, and the tissue healing outcomes. Additionally, they also study how silver is absorbed, distributed, metabolized, and excreted in the body. For clinical evaluation for combination products, it is a multifaceted approach. So, first we have the MOA and intended use, which clearly defines the mechanism, the primary action, and the benefits, making sure that they align with the regulatory pathways, especially when combining components with varying and distinct functions. And the risk benefit assessment evaluates new risks introduced by the combination, including administration methods and efficacy standards. Quality standards adhere to rigorous clinical trial standards, focusing on endpoint driven methodologies and statistical robustness. Added drug benefit and new ingredients demonstrate functional improvement or reduced adverse effect and ensure safety through the pharmacokinetic evaluation and novel components. Regulatory goal establish comprehensive data to support the safe and effective integration while also advancing regulatory science. Regulatory science for combination products. So, first we have to evaluate the complexity by including multiple components like drugs, devices, and biologics. It makes the regulatory process uniquely challenging. But there are certain FDA and other initiatives that focus on prioritizing clear pathways for combination product regulation. So, the core focus and future goals is heavily reliant on developing a life cycle assessment tool and also new standards that also support the growth and development of innovations. So next, we are going to take a look at the new tools and technologies, computer models and simulation. CMS, it plays a vital role in evaluating the combination products. What it does is that essentially it predicts the outcomes and it assesses the product performance in a simulation environment, which is basically a virtual condition. So CMS, it enables us to study the complex interactions, for example, the drug deposition and the flow in the drug eluting stents without actually going for or seeing the need for an extensive clinical trial. By using these models, we can assess the safety and efficacy while also minimizing the trial cost and timelines. So these models they simulate the product behavior and by how we have a standard for ensuring that the simulations produce reliable predictions is that FDA has a program called OSEL, which makes sure the credibility is verified. And the key applications include that the patient simulations are virtual and they cut cost and risk. And performance testing for devices like heart valves also assesses the long term functionality. And also the clinical impact, which enhances safety, supports regulatory decisions, and also improves product design, especially in complex devices. For artificial intelligence, right now it is transforming healthcare. It analyzes real world data to support decision making and predict outcomes. In the case of combination products, AI can optimize the drug design, improve diagnostics, and personalize treatments individually. For example, devices like GI Genius, it is used for early colon cancer detection, which involves the use of AI driven imaging. While Abilify My Sight, it integrates adherence tracking with drug therapy, like a digital pill for tracking medication adherence. So AI is mostly renowned for its predictive capabilities, which complement how we are doing it today, like the traditional evaluation methods. So in this case, we can streamline both the preclinical testing as well as the post-marketing surveillance. Organ on chips. Organ on chips are essentially microfluidic devices and they mimic the human organ functions. So, again, they provide an highly accurate model for studying the drug effects and safety. These chips simulate organ interactions, like, for example, lung airways or when it comes to blood vessels. And again, they are used in preclinical testing. So because these chips they support ADMET profiling and as well as PKPD modeling, they offer a viable alternative to animal testing. And this is beneficial in terms of ethical standards and provides more human relevant results for drug and device testing. And future potential, this can be used in conjunction with computational modeling and AI or machine learning. So this offers precision for testing complex product interaction, and it focuses more on the shift towards predictive and more human relevant preclinical models, which drive safer and faster product development. So this is a diagram that illustrates an interconnection of organ on chips technology, and this simulates a multi-organ interaction like absorption, metabolism, immune response, and clearance. This highlights the drug testing application and shows the pathways for mostly aerosol and oral drugs in the microfluidic system. So, new standards. New standards play a critical role in aligning the global regulatory processes and they ensure consistency, streamline compliance. Frameworks such as ISO 124171, it is designed for vascular device drug combinations and they have right now set the benchmark for safety and performance. But due to the rapid advancement and immense technological growth in combination product development, there is a vacuum and it calls for the need for new guidelines, for new testing methods, and also new specifications that are very specific to these emerging technologies. Standards can either be physical, like for example, reference material, documentary, test protocol, or they can either be data driven and they provide a comprehensive foundation for accurate regulatory assessments and lifecycle management. It is necessary that we expand and keep refining these standards to make sure that we keep up with the innovation while also supporting the patient outcomes and also calling for a global regulatory alignment. So, new methods and approaches. We have new methodologies such as systematic reviews and meta analysis, which provide robust scientific evidence. And what they do is that they consolidate data from multiple studies and eliminate the chances for redundancies and also enhance the reliability of safety and efficacy assessments. These approaches are particularly critical and they are used in preclinical and clinical stages. By reducing redundant studies and offering a holistic overview of product risk and benefits, these methods ensure more efficient regulatory decisions, enabling the development of safer and high quality combination products. Next, we have innovative methods which are streamlining the regulatory and development processes of combination products. So, first, we have evidence based research, which is like a systematic review and meta analysis to again consolidate data and offer a more robust and holistic insight while also eliminating redundant studies. And we have the risk and feasibility assessment, which is a key for again novel drug carriers. These evaluations they make sure that the early stage regulatory approvals and preclinical study designs are streamlined. And we have real world evidence. This essentially leverages the longitudinal and a large sample data set to assess safety, efficacy, and the post market performance, bridging gap between clinical and real world applications. So, as we conclude, we looked at the innovation and complexity. So, advancing the advancements in biomaterials and technologies that are introducing and bringing about new combination products in the regulatory landscape for uh, innovation. So, these products, taking into account how novel and how complicated they are, there needs to be innovative approaches for how we regulate them. And evaluation is carried primarily on the mode of action and the design rationale and the preclinical clinical validations to meet the safety and efficacy benchmarks. With the diverse and the global regulatory variation across countries, often with the differing definitions of PMOA and requirements for complete harmonization, it is coming into light. And the emerging tools like CMNS, AI, ML, and organ-on-chip technologies, along with the very robust post-marketing surveillance, are vital for advancing the safety and efficacy of combination products. So, despite combination products offering tremendous potential for advancing healthcare, there also needs to be a call for advancement in regulatory science, including new standards, tools, and approaches. So we can ensure that these products are both safe and effective. As combination products continue to evolve, regulatory science will play an essential role in guiding their development, making life-changing treatments pivotal and innovative combination products available to patients worldwide. Thank you.
Thank you, Nabila, for the thorough